Welcome back to our bio series. Today's topic is one of the most important but also one of the most difficult. But don't worry, I will make it super easy for you to understand, so stay with me till the end. Because once you get this concept, so many other things in biology will suddenly start making sense. Now let me ask you a question. Have you ever wondered how plants make their own food or how our bodies get energy from the food we eat? The answer lies in bioenergetics, the fascinating study of how energy flows in living systems. Stay till the end, and I promise you will not only understand photosynthesis and respiration clearly, but you will also see how life itself depends on energy transformation. So let's begin with the basics. Bioenergetic is the study of energy transformation in biological systems. Every living cell, whether plant, animal, or microbe, needs energy to grow, repair itself, move, and reproduce. The golden rule is simple. Whenever bonds are broken, energy is released, and whenever bonds are formed, energy is stored. Now the big question, what is the ultimate source of energy for life on Earth? Yes, it's the sun. Although energy comes from sunlight, but all organisms actually rely on chemical energy stored in food molecules. And in living systems, two major processes manage this flow of energy. Photosynthesis stores energy and respiration releases it. Together they form the cycle of life energy transformation. Let's start with photosynthesis. Photosynthesis literally means to produce using light. The word photo means light and synthesis means to produce. So what goes in reactants and what comes out as a product? So the reactants of the photosynthesis is the carbon dioxide, which is taken from the air through stomata, the water molecules which absorb from the soil by roots, and the light energy which absorbed by the chlorophyll and other pigments. And the product of the photosynthesis is the glucose, which is the main sugar produced, which can be stored as a starch or used for energy. And the next product is the oxygen, which releases into the air as a byproduct. Now the overall equation is 6 molecules of carbon dioxide and 6 molecules of water. In the presence of light and chlorophyll produce 1 molecule of glucose and 6 molecules of oxygen. Now think of it like a solar-powered kitchen. The ingredients carbon dioxide and water go into the pot. Sunlight is the stuff heat and the plant's chlorophyll is the chef. The final dish is glucose and oxygen is like the aroma released into the air for everyone to enjoy. So in simple words, photosynthesis is the process by which plants use sunlight to convert carbon dioxide into carbohydrates, specifically into glucose. And the light itself is a form of energy that travels in waves, but our eyes can only detect a small part of it, known as the visible spectrum, ranging between 390 to 760 nanometers. Inside plants, there are special pigments that capture these lights, this specific visible spectrum light. The main one is chlorophyll, which is a complex compound containing magnesium at its center. Now here is a question that why chlorophyll have magnesium at its center? So the answer relies on its role in capturing light energy. Without magnesium, chlorophyll cannot trap light efficiently. So the entire process of photosynthesis slows down or stops. Think of chlorophyll as a satellite dish, but the magnesium atom as the receiver in the middle. Without the receiver, the dish can't pick up signals. Similarly, without magnesium, chlorophyll can't pick up sunlight energy. Now chlorophyll observes mostly violet, blue and red light while reflecting green, and that's why plants appear green to us. Along with chlorophyll, there are accessory pigments called carotenoids such as keratin and xanthophylls, which capture light and range that chlorophyll misses, usually between 500 and 600 nanometers. Now what's the important here is the plant have different types of chlorophyll. Chlorophyll A, B, C, D, E and even bacteriochlorophyll and bacteria. Plants mainly have chlorophyll A and B. Algae may have A, B, C, D or E depending on species, and bacteria have bacteriochlorophyll. And the efficiency of these pigments can be studied in two ways. The absorption spectrum tells us how much light is observed at each wavelength, while the action spectrum shows us which wavelengths are most effective for photosynthesis. In eukaryotes like plants and algae, photosynthesis happens inside chloroplast. Inside chloroplast, you have grana, which is a stick of thylakoid membrane. They look like a pile of coins. These staked membrane hold chlorophyll and other pigments for trapping sunlight. But prokaryotes like cyanobacteria don't have chloroplast. So where do they put their pigments? They spread them out on special unstacked membrane inside the cell called photosynthetic membrane, which is a thin plate membrane floating inside the cell. So in short, in eukaryotes, these pigments are located inside the chloroplast, while in photosynthetic prokaryotes, pigments are found in unstacked photosynthetic membranes. Now let's look at the actual process of photosynthesis. It has two stages, the light reaction and the dark reaction. The light reactions occurs in the grana of the chloroplast. Here, pigments are organized into clusters called photosystems. And these photosystems are the actual working units that capture sunlight. When a photosystem observes light, its electrons get excited and passed along an electron transport chain. As these electrons move, two important things happen. Water is split into hydrogen and oxygen, and energy is released to make adenosine triphosphate ATP. This process of making ATP in the presence of light is called photophosphorylation. There are two types of photophosphorylation: cyclic, which involves only photosystem 1, and non-cyclic, which involves both photosystem 1 and photosystem 2. Now the next is the dark reaction, also known as the Calvin cycle, and it occurs in the stroma of the chloroplast. This cycle was discovered by Melvin Calvin. At this stage, carbon dioxide combines with a 5-carbon compound called ribulose bisphosphate to form a 3-carbon compound called phosphoglyceric acid. But here is a confusion. We know that ribulose bisphosphate is a 5-carbon compound, and when it reacts with a carbon dioxide, which is a 1-carbon compound, mathematically you might think it should make a 6-carbon compound. And actually that's true. Robisco bisphosphate plus carbon dioxide makes a 6-carbon compound intermediate. But here is the key. This 6-carbon compound intermediate is extremely unstable. It doesn't stick around. Almost immediately it splits into 2 molecules of a 3-carbon compound called 3-phosphoglyceric acid. So the confusion clear up Once you realize the 6-carbon intermediate exists but is too unstable to last, it instantly breaks into 2 3-carbon molecules. And this step is called carbon fixation. Then phosphoglyceric acid is converted step by step into another 3-carbon compound called glyceraldehyde 3-phosphate. Now why is glyceraldehyde 3-phosphate so important? And photosynthesis. Glyceraldehyde 3-phosphate is the first stable carbohydrate product. From it, plants can build glucose, starch, cellulose, fats, and even amino acids. Glyceraldehyde 3-phosphate is the raw material for live food molecules. The Krebs cycle ends with the regeneration of ribulose bisphosphate. How ribulose bisphosphate regenerated and how this Krebs cycle ends with regeneration of ribulose bisphosphate. So this is where the cyclic nature of the Krebs cycle becomes clear. Let's walk through it simple. Most glyceraldehyde 3-phosphate stays in the cycle, and this is the key part. To keep the cycle running, the 5-carbon acceptor molecules rubisco bisphosphate must be regenerated. And here is how it happens. Several molecules of glyceraldehyde 3-phosphate are rearranged through a series of enzyme control steps. The rearrangement produces rubisco bisphosphate again. That's why it's called a cycle. It ends where it started, with rubisco bisphosphate ready to capture more carbon dioxide, and the cycle can continue fixing more carbon dioxide. To summarize, the light reaction captures energy, while the dark reaction uses that energy to fix carbon dioxide into carbohydrates. With glyceraldehyde, 3-phosphate is the key building block. But photosynthesis has a problem, and this is where photorespiration comes in. Photorespiration is a process in which oxygen combines with ribulose bisphosphate and the presence of sunlight. Instead of fixing carbon dioxide, the enzyme ribulose bisphosphate carboxylase oxygenase, commonly called rubisco, mistakenly bind oxygen, and this produces carbon dioxide instead of carbohydrates or glucose. Essentially, it's a wasting of energy. Photorespiration usually happens when carbon dioxide levels are low and oxygen levels are high. In short, photorespiration is the reverse of photosynthesis because it hampers carbon dioxide fixation and reduces the efficiency of photosynthesis. Now, nature, however, has a clear solution. Some plants evolved a mechanism known as C4 photosynthesis to minimize photorespiration. In normal photosynthesis, also called the C3 pathway, the first stable product is a 3-carbon compound, but in C4 photosynthesis, the first product is a 4-carbon compound called oxaloacetate. These plants, which include sugarcane and maize, use a special enzyme called phosphophenol pyruvate carboxylase to fix carbon dioxide instead of rubisco. This makes the process much more efficient in hot and dry conditions. So while C3 plants lose energy in photorespiration, C4 plants thrive by avoiding it. Now let's switch gears to respiration, which is the opposite of photosynthesis. Respiration is an oxidation reduction process in which organic food molecules are broken down to release energy. But in anaerobic respiration, which requires oxygen, 1 molecule of glucose produces 36 molecules of ATP. This process occurs partly in cytoplasm and partly in the mitochondria. The first step is glycolysis, which takes place in the cytoplasm. Here, glucose is broken down into 2 molecules of glyceraldehyde 3-phosphate, and then each of these molecules is oxidized to form pyruvic acid. The net gain in glycolysis is 2 ATP. This step is common to both aerobic and anaerobic respiration. Now here is something interesting. Notice how glyceraldehyde 3-phosphate shows up again. In photosynthesis, it is a building block for making glucose, but in respiration, it's a breakdown product of glucose. That's why glyceraldehyde 3-phosphate is called a central hub compound that connects both processes. Then after glycolysis, pyruvic acid is converted into acetyl coenzyme A, acetyl CoA, before entering the Krebs cycle. And in the Krebs cycle, which takes place in the mitochondria, acetyl CoA is completely oxidized. And this releases carbon dioxide, which reduces nicotinamide adenine dinucleotide NAD to NADH2 and flavin adenine dinucleotide FAD to FADH2, and also produces ATP through substrate level phosphorylation. The final stage is the electron transport chain, where NADH2 and FADH2 donate electrons to oxygen. So oxygen is the final acceptor of the electron in the electron transport chain, and this step produces the majority of ATP. Now if oxygen is absent, there will be anaerobic respiration or fermentation, and this is incomplete breakdown of glucose. In human muscles and some microbes, glucose is converted into lactic acid and to ATP, and that process is known as lactic acid fermentation or lactate fermentation. And in yeast and some organisms, glucose is converted into ethanol, carbon dioxide, and to ATP. So that process is known as alcoholic fermentation. So now you can see that life on Earth is basically a game of energy transformations. From the sunlight captured in photosynthesis to the energy lost in photorespiration, to the adaptation of C4 plants, and finally to the ATP released in respiration. All of it revolves around key compounds like glyceraldehyde 3-phosphate, which link both processes together. In our next lecture, we will solve tricky MCQs based on bioenergetics, where you will see how examiner twists these concepts into questions and how you can master them easily. Don't miss it. So this was the most important and also the most confusing topic. If you still have some confusion, so you can drop your question in the comment section. Now, if you found this helpful, don't forget to like, subscribe, and share with your friends. Until next time, keep practicing and keep learning.